astrocytes and microglia. Now back to something completely different. I'll try and tie everything back together at the end, but we have to talk about glial cells now. Okay, well, we're going to come back and look at propagating action potential with myelin, and myelin is put on by some glial cells, as we're going to find out. Okay, astrocytes and microglia. Okay, this is my standard representation of a neuron. Right here's your dendrite, your cell body, axon hillock, axon terminal. Okay, and these are both found in the CNS. These are astrocytes and microglia are both in the central nervous system. This long structure down here is just my representation of a blood vessel. Okay, get a little red for color. So this can be any cranial artery. Let's just label it as a cranial artery, a blood vessel. Okay. In general, we're going to find out the glial cells are supporting cells. In the central nervous system and the peripheral nervous system, you either have neurons or you've got glial cells. It's one or the other. Okay, not much more than that. Okay, here we have a picture of your basic neuron, and then this is a blood vessel. I'm going to start out talking about astrocytes. Okay, astrocytes. Okay, astrocytes, as the name sounds, they're star-shaped cells. So to start out drawing the astrocyte, I'm going to put it here, and the astrocyte has these appendages that stick out. They're kind of like little feet. And I'm going to draw some little feet sticking out of this star-shaped cell here. I'm going to put 3 feet attaching to the soma of this neuron. Okay. And then I'm going to have 3 feet down here. And these feet I'm going to draw so that they're juxtaposed. Okay, 1 foot there. And what I want to show is that there's like a tight connection between the feet that I'm going to draw that are attached to the blood vessel. Got my 3 feet there. And I'm also going to have 2 feet just sticking out here in space. And this is going to be your astrocyte. It has a nucleus. It's the most predominant glial cell is going to be your astrocyte. We'll hear this. This astrocyte. The most predominant glial cell. Okay, and they are a star-shaped cell. Hence the name astrocyte. Bless you. Okay, there's also 4 different functions that I'd like you to be aware of for the astrocyte. I'm going to label them as we go. So these connections here, I put these feet with kind of tight connections for the astrocyte. You can see they're just on top of each other. and they're juxtaposed, they're attached to the blood vessel. Okay, and that one function of the astrocyte, or the first function I'm going to talk about, is supporting the blood-brain barrier. Okay, essentially these guys are in charge of making sure that things that you don't want in the CNS don't come in from the blood vessel. Okay, it's going to be the first function of the astrocyte I'd like to consider. Okay, function number 2. It's going to be here. Okay, and if you can imagine, in the blood vessel, the major fuel for your brain is going to be glucose. It prefers glucose. It can use ketone bodies if absolutely necessary, but it definitely prefers glucose. And let's just consider glucose a basic food. Okay, so floating around in your blood is food, nourishment. Okay, one function of your astrocyte is going to be to take up food source and relay it to the neuron. So in food here, you can think of glucose, or it's going to be your predominant food. So first, it's going to help support blood-brain barrier. Second, the astrocyte's going to help nourish the neurons by taking up food from the blood vessel. Okay. Also remember, when we were propagating this action potential, you'd have the uptake of sodium, but after the sodium channel's closed, you have that release of, what was that other? Potassium. Right, so you have the release of potassium into the system. So let's go ahead and say we have increase in potassium due to action potentials. Okay, a third function for the astrocyte is going to be taking up this extra potassium and help neutralize the extracellular environment. Okay, first function, support that blood-brain barrier. Second function, help provide food or nourishment to the neurons. Third function, take up that extra potassium that exists in the extracellular space. Okay, and then there's one more function. Where is it? The fourth function here is going to be release neurotrophic factors. Okay, releasing neurotrophic factors. Does anyone know what a neurotrophic factor does? Any thoughts? And these are factors, or yeah, if you want to think about chemicals, they're chemicals released by the astrocyte that are going to help guide neuronal growth. Okay, so it turns out that the nervous system itself is very plastic. It can change. Way back when they used to think it was set, can't change it. Once you reach a certain age, nothing changes. Now we know there's something called neuroplasticity, and that's what we use when we're working with patients that have had neurological injury in neurorehab, right? Something called neuroplasticity. Things like use it or lose it, intestinal matters, salience matters, lots of different things that you'll learn. So it turns out that if there's axonal damage that happens in the central nervous system, the astrocytes actually release neurotrophic factors that cause the regrowth or the guiding of those axons to where they need to go. Okay, so it's a neuroplastic process. And that's what neurotrophic factors are. Okay, astrocytes in general, star-shaped cells, have 4 different functions that you need to remember. They support the blood-brain barrier, they provide nourishment to the neuron, they take up extra potassium from the extracellular space, and they help release neurotrophic factors that help guide axonal neuronal growth in response to injury. Okay, questions about that one? Okay, and your astrocytes, they're going to be your most predominant glial cell. Okay, so most of those are in. Then think about microglia. Okay, thinking about microglia, the word microglia, what are these things going to look like? They're really small. Yeah, they're teeny tiny, essentially. And as far as microglia are concerned, well, you guys know what this is. Let's see. If I drew something like this, what does that look like? Does anybody know any? Looks like a virus. That's right. It is a virus. It's my rendition of COVID. Okay. So this is let's just say these are viral particles. Okay, your viral particles. Somehow a virus got into the central nervous system. And we do know that happens with COVID, right? You've heard of long COVID and that sort of thing. So viral particles got in there. Now your nervous system has to have a way to deal with these viral particles. And how they deal with it is by using the microglia. Okay? Microglia themselves are small in comparison to the astrocyte. They're very small. And they're going to kind of look like this, and they've got some little fringe on the outside. Okay, and these are your microglia, and they function as the immune system for the central nervous system. So let's just say CNS immune system. Their job is just going to be to recognize and get rid of foreign invaders. Microglia, in general, they're kind of glial cell. They're really small. They function as the CNS immune system. Just another kind of glial cell. Okay, astrocytes and microglia are only found in the central nervous system. They're not peripheral nervous system. Any questions about that? 2 different kinds of glial cells. We're going to talk about a few more as we go. Good? All right, let's move on to appendable cells. Okay. That's my standard frontal section. Okay, you have your cortical region, midbrain, pons, medulla, spinal cord. Does anybody remember what these things are called? These are nuclei inside. Thalamus, yeah, those are my thalamus. I'll label that as thalamus. Okay, a little bit of a review from the other day. We have this ventricle that's here, resides between the thalami. Okay, does anybody remember the name of that ventricle? That's my 3rd ventricle. Great. And connecting the 3rd ventricle to these pair of ventricles here on the side, I have a special kind of foramen. Does anybody remember the name of that foramen? It's foramen of, anybody? Monroe. Okay, off of the third ventricle, connecting the third ventricle to your lateral ventricles, that's going to be your foramen of Monroe. Okay, foramen of Monroe is going to be on the very top, and its job is going to be to connect third ventricle to your lateral ventricles. I'll just draw those out here. Okay, so third ventricle, pair of lateral ventricles on the side. Okay, underneath, at the very base of the third ventricle, there's a connector that goes through the midbrain. Does anybody remember what that connector is called? Cerebral aqueduct. Oh, good. You got it quick this year. Cerebral aqueduct. Okay. And then your cerebral aqueduct is going to go ahead and connect down here to your fourth ventricle. And your fourth ventricle is going to go ahead and connect down to your central canal. Goes all the way down the spinal cord. Okay, here's your fourth ventricle. Here's your cerebral canal. No, spinal canal. Central canal. There's your central canal. Okay. Again, small review. What are the names of the foramen on the outsides? Yeah, foramen of Volushka. Okay. Wuschka, and then there's one in the back, in the middle. Magendie, yeah, foramen of Magendie. I like repetition and review. It's just easier to remember things if you do it multiple times, I think, anyway. Now, on to the subject of this slide, which is ependymal cells. Okay, what do ependymal cells have to do with this? Well, ependymal cells are nothing more than your basic columnar cell here, and your ependymal cells line all of your ventricles. Okay, so your basic, it's a columnar, looks like a column standing up. Okay, and these are your ependymal cells. Okay, basic columnar cell is going to line all of your ventricles, and for the purpose of brevity, I'm just going to line this lateral ventricle, so they line all ventricles. Okay.
There would be a space for there.
I'm going to fill that space in a minute. But we talked about a special, so there is a specialized type of ependymal cell, and that specialized type of ependymal cell is responsible for making CSF. Can anyone tell me what the name of that one was? Choroid plexus, right. So the choroid plexus, I'm going to make those in here. This is just a specialized, and I think I had a little squiggly on the outside of these are my choroid plexus. They're nothing more than a specialized type of ependymal cell. And these choroid plexus actually work by filtering blood flow and pulling out cerebral spinal fluid, which is, and secreting it into the ventricular space. So this is your choroid plexus. And that makes CSF. Okay. So choroid plexus is nothing more than a specialized kind of ependymal cell. One more thing about these ependymal cells, they all have little cilia associated with them. Does everybody know what cilia is? Yeah, it's like a little hair that can wave back and forth. Why would it be important to have the ependymal cells have a cilia? Any thoughts? Yeah, it helps mobilize the CSF through the ventricular space, right? Rachel's got it down. She's got this kind of motion going, right? Okay, so they're just going to wave back and forth, and you're going to help the flow of CSF through the ventricular space. Questions about that? So ependymal cells, they're a kind of glial cell. Basic columnar epithelia, they're actually ciliated columnar epithelial cells. The specialized type of ependymal cell is known as choroid plexus, and that's the one that makes your CSF. We talked about that one before. Got a little bit more of a review for our ventricular system, as well as you should know that ependymal cells line all of the ventricles. Question, Tyler?
No.
Oh, that's okay. Feel free to stretch. Anybody have any questions? This is pretty straightforward in ependymal cells, and really it's, okay. The other thing that you need to know about ependymal cells, again, they're only in the CNS. Okay, a little bit of a review, a little bit more about the ventricular system. Has to do with the glial cell. Ependymal cells go with glial cells, ventricular cells. Okay, now we're going to get to the myelinating glial cells. We talked a little bit about myelin before and its importance in helping the speed of transmission of an action potential down the axon. So, myelinating glial cells. We have two different kinds. There's going to be one kind that's only in the CNS. Okay, I'm going to draw that on the left-hand side of the picture. And then there's going to be another kind that's only in the PNS. I'm going to draw that on the right side of the picture. I'm going to start with the CNS over here. And the myelinating, does anybody know the myelinating glial cell in CNS? It's got a cool name. I think it's cool. It's an oligodendrocyte. Oligodendrocyte. I have a good friend who had a cat, and she named him oligodendrocyte. He was called Ollie for short. Says a little bit about my friends, maybe, I don't know. she's great. Yeah, Ollie unfortunately passed on. But oligodendrocytes, they're the myelinating glial cells, only in the central nervous system. And the myelinating glial cells, the oligodendrocyte, so what they're going to do is wrap the axon of the neuron. And I'm going to try and, I'm going to try and draw the wrapping so it's like a little swirly here. So that means it's wrapped around the outside of the neuron, okay? And that's going to be one. That's going to be one myelin sheath coming for the oligodendrocyte. And then the cool thing about oligodendrocytes is they can put out multiple appendages and make multiple myelin sheaths on individual neurons or even adjacent neurons. So they're really not limited as far as their scope is concerned. Okay. So here I have 3 myelin sheaths that are being put out by the oligodendrocyte, and the oligodendrocyte itself has a cell body. Puts out these appendages that wrap the axon with myelin sheaths. Okay, so this thing is a myelin, M-Y-E-L-I-N, sheath, S-H-E-A-T-H. It's a myelin sheath. So there's one oligodendrocyte, it puts out 3 appendages, or many, many appendages, and one cell is capable of providing, one oligodendrocyte is capable of providing myelin sheaths, multiple, to either the same neuron or even other adjacent neurons. Okay, so it's not really limited. This one cell is capable of producing many, many myelin sheaths. Does anybody know what the disease process is caused by disruption of myelin sheaths in the central nervous system? MS, yeah, multiple sclerosis, good. We're going to talk about that here in a minute. Okay, oligodendrocyte, only in the CNS. In the PNS, we have a different cell type. Does anybody know that one? Schwann cells, yeah. Schwann cells, S-C-H-A-W, Schwann cell. C-H-W-A-N-N cell. It's a Schwann cell. Okay, Schwann cells are found only in the PNS. Where's my, choose this one. Okay, thinking about Schwann cells in the PNS, it's a little bit different. Here you have, so the same deal. I'm going to just wrap this myelin sheath around the axon. Okay, here. But this whole myelin sheath is the Schwann cell. Okay, so that's the cell nucleus. So in contrast to the oligodendrocyte, they can put out multiple myelin sheaths per neuron. The Schwann cell itself wraps itself around the axon, and that's one whole cell. Okay, one whole Schwann cell. So these are 3 different Schwann cells. I'll label that here. Okay, Schwann cells are found only in the peripheral nervous system. And it takes a whole Schwann cell to just produce one little segment of myelin sheath wrapped around an axon. Questions about that? Pretty straightforward. Okay, it's just myelinating glial cells, 2 different kinds. Oligodendrocytes specific to CNS, Schwann cells specific to PNS. And then there's one more kind of cell that helps support neurons in the PNS. It's only in the PNS. And I'll just draw a little, this is just a supporting cell. It's called a satellite cell. Okay. And it does nothing more than support neurons in the PNS. An easy way to think about a satellite cell would be similar to an astrocyte, okay, providing nourishment and helping things along. Okay, sucking up maybe some extra potassium, that sort of thing. But the satellite cell itself, only in the CNS, or the PNS, not in the CNS. Okay, so specific to the PNS, you have your Schwann cell, your satellite cell. All the others are just in the CNS. Questions? Okay, we'll move on. Last one. Neuron communication with and without myelin. Okay, for this one, I'm going to choose to draw some, I'm just going to say Schwann cells, okay, just because they're easier to draw than the oligodendrocytes, I think. I'm just going to draw 3 Schwann cells here wrapped around the axon of this neuron. So this is just 2 neurons in sequence, dendrite, soma, axon, synaptic terminal. So here's 1 Schwann cell. Okay, another Schwann cell. And then the 3rd Schwann cell. Okay. And the same thing happens in the CNS. I'm just choosing to draw Schwann cells because I think they're easier to draw right now. Okay, 3 different, I'll label those as, these are just Schwann cells. If it were in CNS, it'd be oligodendrocytes. Okay, now we talked about this a little bit before. We talked about the mechanically gated ion channels causing the influx of sodium into the cell. And when that sodium threshold reaches minus 50 millivolts, and that signal is received by the axon hillock, you have the action potential starting. All right, so the action potential starts here. So that is your action potential. And that's your axon hillock. Okay, minus 50 millivolts is received by the axon hillock, and the action potential happens. And in the presence of a myelin sheath, here we have 3 different ones formed by these Schwann cells, the action potential can just skip right over the Schwann cell or the myelinated area and propagate at the next available space. Okay, and again, it can skip over the Schwann cell, the myelin, and propagate there. So you can see signal transmission is going to be a whole lot faster in the presence of myelinated axons. And there's a couple of terms that you need to be aware of. This jumping, so like when the action potential jumps from one thing over top of the myelin sheath to appear where there's an empty space that isn't myelinated. Does anyone know what that's called? Has anybody heard of that word before? It's called saltatory conduction. S-A-L-T-A-T-O-R-Y, conduction. And saltatory conduction means nothing more than the action potential is able to skip over the myelinated areas of the axon. And these non-myelinated areas of the axon have a special name. Anybody know what that is? No. Okay. Yeah, these are called nodes of Ranvier. Nodes of R-A-N-V-I-E-R. Nodes of Ranvier. Okay, so having a myelinated axon helps the action potential propagate faster because due to saltatory conduction, that action potential can jump from one node of Ranvier to the next, go a little bit faster. Questions? No? Okay, let's talk about some stuff. We talked about, or I asked a general question, in the central nervous system having a demyelination problem or a problem with demyelination. Talk about that being due to multiple sclerosis. Okay, so in this multiple sclerosis is a demyelination in the CNS, central nervous system. That's brain or spinal cord. Okay? It's called multiple sclerosis if it's appearing in your brain. It's actually called transverse myelitis if it's appearing in your spinal cord. Minor difference, it's the same thing, only minor difference. Brain versus spinal cord. We'll talk more about that next year. For right now, you need to know multiple sclerosis. Okay, multiple sclerosis is demyelination in the CNS. Does anyone know anyone with multiple sclerosis? Yeah. Wow, quite a few. Okay. So the target cell here, because it's in the CNS, what's the target cell that's affected? What cell is oligodendrocyte? Right. Oligodendrocyte. Dendrocyte has been affected in multiple sclerosis. Okay. And the problem with multiple sclerosis is that the signs and symptoms are really variable from one patient to another. And oftentimes the signs and symptoms are actually, they're more symptoms than signs. What's the difference between a sign and a symptom? That's a board's question. It's a good one. Anybody know what's the difference between a sign and a symptom? Any thoughts? Anybody heard about that before? Sorry?
Signs might not bother you.
I'm sorry?
Do signs might not bother you, whereas a symptom like affects your brain.
Anybody? Matt, did you have a thought? You're just stretching too. Okay. So a sign is going to be something I could read. How about that? So a sign would be increased heart rate, increased blood pressure, right? Something I get, something that I can see, something that as a therapist I can see. That's going to be my sign, right? A symptom is going to be like fatigue, right? So patients with multiple sclerosis typically experience tremendous fatigue. Okay, but they go to the doctor and they say, well, I'm just really tired all the time. So, are you sleeping? Well, yeah, I'm sleeping, but I'm really tired. And my vision is just blurry. I don't know why. Or I feel like pins and needles in my arm. This like paresthesias, right? I just feel this tingling sensation in my arm. Or, I feel dizzy. These are just symptoms that a patient might complain about. Okay? Signs and symptoms are going to be variable. And it is a lifelong problem. A lot of interesting research being done right now with multiple sclerosis. We'll learn about that next year. Okay, in the PNS, what's the demyelination problem in the PNS called? Does anyone know? Anyone heard of that before? Yep, Guillain-Barré. Guillain-Barré syndrome. Another thing you need to know, so multiple sclerosis is actually autoimmune. Nobody really knows what causes it, but it is definitely an autoimmune problem. Okay, your body's attacking cells. Guillain-Barré is also an autoimmune problem. Guillain-Barré, it's usually it presents after a viral infection of some sort. Patients can present with sensory changes as well as weakness. A couple weeks after their infection, it usually starts distal and then proceeds proximal. Okay, it can be so bad that the person requires a ventilator just to breathe. Okay, so serious, serious weakness. So the person gets these problems, it reaches a peak, and then it starts to go back down. It's a problem with Schwann, so your immune system's attacking your Schwann cells. Okay, so it's demyelination of your peripheral nervous system. Weakness, sensory changes, overall severe weakness. But it's 80 to 90% of the individuals recover within 2 years. Okay, so that's not everybody, right? There's like 10 to 20% who don't fully recover within 2 years. And that's a pretty big difference. One more thing you need to know about a demyelination disorder that happens specifically in the peripheral nervous system. This is a weird one. It's called Charcot, C-H-A-R-C-O-T, Marie-Tooth syndrome. Okay, where these two were autoimmune, this one's genetic. So you're born with Charcot-Marie-Tooth disease. And specifically, it's a mutation for a kind of protein called connexin. You don't really have to know that, but it's a kind of protein that's really important for Schwann cell integrity. Okay, patients are going to present with decreased strength and sensation, specifically in their lower extremities. And it is a progressive problem. It gets worse as you get older. I've seen patients in the clinic with Charcot-Marie-Tooth. So this is the patient population that we want to try and compensate with, so get them AFOs and teach them to walk with a canter walker. Yes, Trinity?
Do they normally find out sooner, like they get tested on it, or is it just later in life they learn about it?
Yeah, I don't know that you get tested for it, but if it's inherited, I don't know what the inheritance pattern is. That's a good question. Yeah, I don't know what the inheritance pattern is. If it's autosomal dominant, then you'd expect one of your parents to have it as well. I don't know the answer to that question, but I do know, I don't know when signs and symptoms start to present either. But just, you know, the individuals that I've seen in the clinic for it were in their 40s. Okay, so because it's progressive sensory and strength changes, it takes a while for them to get there. Okay, any other questions? Okay, let's go ahead and